In this video, we're gonna talk about ligand substitution. Now, if that is the case, we are going to talk about ligand substitution to make this thing here, to make this big complex here. You see it's 2+, and it's got 2 waters, and it has got 4 ammonias. So if we look here, there's 2 equations, and I'll go through those 2 equations in a minute. So you've got to add ammonia to make this, and you've got to add it to a complex, and the complex is this, and it's a lovely pale blue colour, and you can see the solution here. So that's what the solution would look like, this lovely pale blue. Now it's got 6 ligands, 6 H2O ligands, and you add ammonia to it. Now, what you make is copper hydroxide, which is this, okay, and it's a blue precipitate. Now, the question is, why do you make copper hydroxide even though you're adding ammonia? Well, that's because the ammonia, when added to water, is going to make ammonium and hydroxide. That's because the ammonia acts like a base and accepts a proton, and it takes the proton off the water, leaving it with OH-. That's why you can do one of 2 equations. So the best one to do is this one down here, and I'll just talk about that one in a minute. But if you're just given Cu2+ and told Cu2, you're not given this, you're given Cu2+, then it's this equation all day long. So you add ammonia to it, and instead of adding ammonia, you're going to add hydroxide, because it makes hydroxide. And that's going to make this here. This is a precipitate. This is a precipitation reaction, essentially, if you ever have to do this. Now, the one underneath is a ligand substitution reaction. So the ligand substitution reaction, you're given this in the question, okay? And you add the 2 hydroxides, and the 2 hydroxides will kick out and substitute 2 of the waters. So 2 of the waters are going to fly off and be kicked out. You can see that there. Okay, and so you make this. So you've swapped out 2 of the waters. You've cancelled out the 2+ charge because you've added 2 OH-. Okay, and this is your full equation you've got to remember. Now, this is with a little bit of ammonia. We keep on adding ammonia, and it stops acting like a hydroxide, and it starts doing ligand substitution. So let's just remind ourselves, first of all, we went from the ammonia and this complex to give copper hydroxide, the blue precipitate, and that's your blue precipitate there. Then if we add more ammonia, we're going to get a deep blue colour. And you can see here that we've got more happening. Remember, it starts as copper with 6 waters, and we've substituted 4 of those to get this complex we have up there. And notice we've got a deep blue colour now as well. And that's what it looks like down there, a very deep blue colour. So here's our equation that we need to remember. And this is basically saying, look, we go from here and we add too much. We add loads of ammonia, which means we'll substitute 4 of them out. We substitute 4 of them out, we get this deep blue complex, and we kick out 4 waters. Okay, so pale blue to deep blue. So there's a difference between a little bit of ammonia and a lot. Okay, and make sure you know both. Next one we're going to do is ligand substitution to make CuCl4, so copper chloride. Okay, now copper chloride is yellow, and the copper hexaqua, we call it here, is pale blue. And what you do is you add chloride to make this copper chloride. Now, the chloride comes from concentrated HCl, and you need to remember it's concentrated HCl, and you need to know that these 2 react. I'm going to show you, this is me in the lab, that's my finger. Okay, and you can see it's green because yellow and pale blue will be green. But I'm keeping on adding the concentrated hydrochloric acid, and you notice it's going more and more yellow. It's going more and more yellow, more and more yellow. So you could get asked the question, how can you make it go yellow? Well, you've got to add more of this, and then the equilibrium will go to the right if you've done your equilibrium. And there's your nice yellow solution. Now, the most likely question on here is, why does this happen? Okay, it happens because of 2 things. The water and the chloride. The chloride is bigger and it is negative. So it's bigger and it's negative. Well, that means you can't fit 6 chlorides around it because they repel each other. So they're charged, they repel each other more than the water. They're larger than the water molecules, okay? So the shape changes from octahedral here, 6 electron pairs that repel each other as far apart as possible, 6 bonding electron pairs, and then to this, CuCl4, which is 4 bonding pairs that repel each other as far apart as possible. So we're going from octahedral to tetrahedral. And that's how you do ligand substitution, because remember you're substituting the chlorides for the water, so it's a ligand substitution reaction And you will be asked what type of reactions these are. So make sure you know. So in this one, we're going to do another ligand substitution, but this time to make chromium hexaamine with a 3+. Now the 3+ is outside the bracket. Inside the bracket is 6 NH3s. They're all neutral, which means it's 3+ outside. Chromium must be 3+. Okay. Now we're adding ammonia, a little bit of ammonia again, to make this. This is violet. Now just warn you, in our video that we've got here, it's not violet because we didn't have any violet versions of chromium in the lab. Okay, but all the rest of the colour changes are absolutely spot on. Now, if we look here, we've got chromium hexa aqua, which is violet again. I'll say violet again. Give a chromium 3 hydroxide. Now, it is acceptable. Let me get out of the way. It's acceptable to have chromium 3+ and just have chromium 3+. They don't mind if you don't use the full equation here. Just use Cr3+ plus 3 of those OH minuses goes to our grey-green precipitate, which is this one here. Okay, and you'll see it coming up here. So we're adding the ammonia here, and you can see this grey-green precipitate just coming here. Now what we're going to do, we're going to add too much in a second, and that grey-green precipitate will disappear. Okay, and you'll get a different colour solution. You see here? Right, okay, so it's starting to disappear. Now, you go from this violet to this grey-green precipitate. Okay, and then when you add excess ammonia, you can see here, okay, you get a purple solution. Now, the one there, as you can see, didn't give a purple solution. Again, we didn't have that in the lab. But you can see the grey-green precipitate. So, if we go from here, excess ammonia to give that purple, give the purple CrNH3 solution, and it is this one instead. So we have got chromium with the 6 aquas, and then basically you've got the 6 amines, replaces all of them, kicks out all of the waters. And you're going from a violet to a purple solution. Sorry, we didn't have the one in the lab to show it to you. Okay, now, you look at that, your colour change will be going from violet to purple, and in the meantime, in the middle of it, you'll get a grey-green precipitate. And that is how you get chromium complexes to be substituted. Next one is blood. You can see all the blood here now. Most of what I'm going to tell you you don't need. I don't want you to switch off, but you don't need. Okay, so what you've got here is your blood cells. Okay, and we've got globin here. So this is hemoglobin. Notice the oxygen is bonded to the iron. Now, the things you need to know is that is a dative covalent bond or a coordinate bond. So it's a coordinate bond. And this hemoglobin carries the oxygen and it does gaseous exchange. Okay, and you've got the 4 nitrogens as well, and you've got the protein globin. There's a coordinate bond forms to the oxygen. Okay, and it also forms to carbon dioxide. Okay, and it gets gaseous exchange. So in your body, it will swap, the carbon dioxide will swap with the oxygen, and so you take in the oxygen, you give out the carbon dioxide. Now, what you really need to know is this, is the bond with carbon monoxide. The carbon with monoxide, and this is your exam answer. Carbon monoxide irreversibly bonds to the hemoglobin, and it blocks the oxygen uptake, and therefore you will be in serious trouble if you don't get medical help or die. And the key thing you need to know for the exam is CO has a stronger dative bond than the bond to the O2. Okay? Now, that is the answer for the question, and that's really all you need to know about this. Now, this video has been about ligand substitution, and it's all about those colours and those equations. So 3 types of questions you're going to get. 1, can you name what type of reaction it is, and it's ligand substitution. 2, can you do the equations? 3, can you do the colours? So there are 3 you need. As I always say on this, like, share and subscribe. Most important thing is share. Okay, and I really appreciate all your support.